Breakthrough. Scientists map genetic resistance to wheat cancer. Imagine a world where our staple food, wheat, is under constant threat from a relentless enemy, a disease so devastating it's often called wheat cancer. But what if we could finally turn the tide? Chinese scientists have just unveiled a groundbreaking genetic map that could be the key to combating the yellow rust disease, a pathogen that mutates rapidly and causes roughly 10% of global wheat yield loss each year. This breakthrough promises not only more durable wheat resistance, but also a significant decrease in pesticide dependency, a critical step towards sustainable agriculture. But here's where it gets controversial. The rapid mutation of the yellow rust pathogen means that even resistant wheat varieties can become vulnerable within just a few years. This makes breeding resistant cultivars a never-ending challenge. Imagine being a farmer, watching your wheat fields turn yellow overnight due to this relentless disease. It's a nightmare scenario, but Chinese researchers have just made a significant leap towards a solution. The research, published in the prestigious Nature Genetics journal, was a joint effort between the Northwest Agriculture and Forestry University, NWAFU, and the Chinese Academy of Sciences Institute of Genetics and Developmental Biology. It's not just a map. It's a powerful tool that provides breeders with unprecedented insights into the genetic makeup of wheat's resistance to yellow rust. To understand why this is such a big deal, think of it like a treasure map for wheat breeders. This map helps them navigate the complex world of wheat genetics to find the best traits for resistance. But this is the part most people miss. The map doesn't just show where the resistance genes are. It also reveals how they interact with the yellow rust pathogen over time. This co-evolutionary dance is crucial for developing long-lasting resistance. The team spent 5 years analyzing data from 2,191 wheat accessions worldwide and over 47,000 records of yellow rust response. Their findings? A whopping 431 yellow rust resistance loci, which they used to create a comprehensive map of yellow rust resistance genes. Among their discoveries, 3 novel resistance genes stand out. Gene YR5X shows resistance to multiple pathogen races. Gene YR6 PM5 offers dual resistance to yellow rust and powdery mildew, and gene YrKB provides broad-spectrum rust resistance without sacrificing yield. The research team believes that these findings could significantly extend the lifespan of resistant wheat varieties, potentially up to a decade or more. This is a game changer for global food security, offering a robust scientific solution to reduce pesticide use and combat this persistent threat. What do you think? Could this breakthrough be the turning point we need in the fight against wheat diseases? Share your thoughts in the comments. Unlocking yellow rust resistance in wheat, a genomics-driven approach. The exploration of genomics has led to the identification of exceptional alleles and genes that confer resistance to yellow rust in wheat, a significant threat to global food security. Data accessibility. The foundational sequence data for whole genome sequencing and RNA sequencing presented in this study has been archived in the National Center for Biotechnology Information, NCBI, under project numbers PRJNA 125391 and PRJNA 1256661. These datasets are publicly accessible at the NCBI website. Additionally, the data has been stored in the Genome Sequence Archive at the National Genomics Data Center, which is part of the China National Center for Bioinformation and the Beijing Institute of Genomics, Chinese Academy of Sciences. The specific accessions include CRA 00432, CRA 041998, CRA 005878, and CRA 021484, all of which can be found on the NGDC website. Furthermore, genotype data in the varied call format, VCF, can be accessed under the accession number GVM 00830. This genotype information is also available through Zenodo. To streamline access and enhance the breeding process for stripe rust resistance, we have created a specialized database dedicated to this purpose. All cloned genes, including PQ112656 /YR1256, previously known as YR5C, PQ112657, YR6, and PQ112658 TAEDR2B, are listed in the NCBI GenBank. Source data accompanying this paper can be found in the provided references. Software availability. All software utilized in this research is readily available online as detailed in the method section and the reporting summary. References. FAOstat Crops and Livestock Products, Food and Agriculture Organization of the United Nations, FAO, accessed 12 January 2024 as https://RTM, FAOstat data-Q/2024. Savary et al. The Global Burden of Pathogens and Pests on Major Food Crops. Not Ego Evil, 3, 430-439, 2019. Hafiz, A.N.M. et al. Creation and Judicious Application of a Wheat Resistance Gene Atlas. Multiplant, 14, 1053-1070, 2021. 4. Hovmuller M.S., Walter and Justison A.F. Escalating Threat of Wheat Rusts. Science, 329, 369 in 2010. 5. Hovmuller M.S., Thackty and Justison A.F. Global Dispersal and Diversity of Rust Fungi in the Context of Plant Health. Current Opinion Microbial, 71, 10243, 2023. Wellings, C.R., Global Status of Stripe Rust, a review of historical and current threats. Euphytico 179, 129-141, 2011. 7. Chen, X, Pathogens which threaten food security. Puccinius striformis, the wheat stripe rust pathogen. FoodSec 12, 239-251, 2020. 8. Beto, J.M. et al. Research Investment Implications of Shifts in the Global Geography of Wheat Stripe Rust. Nat Plants 15142 and Mardid 15. 9. Bouvet et al. The Evolving Battle Between Yellow Rust and Wheat, Implications for Global Food Security. Thier Applegenet 135, 741-753, 2022. 10. McIntosh, Lauren Mu, Handy and Kangzi, Wheat Stripe Rust Resistance Gene YR24, YR26, a retrospective review. Krobj 6, 321-329, 2018. Acknowledgements. We would like to express our gratitude to Prof. Chung Tha Wang for his invaluable contributions to our resistance breeding efforts. We also extend our thanks to the dedicated members of the Plant Immunity Research Team at the State Key Laboratory of Crop Stress Resistance and High Efficiency Production at Northwest A&F University for their insightful feedback. Special thanks to M. Wang, Ji Li, W. Guo, Ji Li, and C. Jiang for their constructive suggestions and discussions. We appreciate R.A. McIntosh for his assistance in language editing and proofreading the manuscript. Additionally, we thank J. Guo, Xbai, and X Huang for their help with genetic transformation, and X Huang, Q Zhang, and X Zhao for conducting RT-qPCR assays and AQP genotyping. We are grateful to H. Now and F. Yuan for their support in confocal experiments, and to H. Wang for constructing the genomic DNA library. We also acknowledge J. Tong for sharing critical YR resistance 2CL information, and J. Du, Y. Peng, L. Zhang, W. Zhang, Zlex Yu, Y. Yang, B. Cheng, and Q. Yao for their assistance in phenotyping. Finally, we thank the high-performance computing platform of Northwest A&F University for providing the necessary computational resources. This research was funded by the National Natural Science Foundation of China and various other grants aimed at enhancing agriculture resilience.